So here's a summary of basically what we've produced by breaking down that 18 carbon fatty acyl-CoA molecule in the beta oxidation. And again, this 18 carbon fatty acyl-CoA molecule is gonna require 8 turns of that beta oxidation process in order to fully break it down into that 2 carbon acetyl-CoA molecule. And again, the number of turns of beta oxidation to break down fatty acyl-CoA is going to be determined by the number of carbon atoms in that fatty acyl-CoA molecule. So again, the more carbon atoms you have in the fatty acyl-CoA molecule, the more turns of beta oxidation is going to take to completely break it down. Because as I mentioned, each time beta oxidation takes place, we cleave off 2 carbon atoms. So for an 18 carbon fatty acyl-CoA molecule, once it's fully broken down through beta oxidation, we've produced 9 acetyl-CoA in total. So 1 for every turn of the beta oxidation cycle, and then also that 1 acetyl-CoA that we're left with it at the end. So that's 8 plus 1, 9 in total. And then we've also produced 8 NADH and 8 FADH2. And again, the number of NADH and FADH2 is simply the number of times beta oxidation takes place for a given fatty acyl-CoA molecule. And to determine the number of NADH and FADH2 to produce for a given fatty acyl-CoA molecule, you simply take the number of carbon atoms within that fatty acyl-CoA divided by 2 and subtract 1. So again, here we have that metabolic kind of master map of all our metabolic pathways that we're gonna look at. And now we can add the beta oxidation to this slide. So again, we start off with triglycerides. We broke them down into free fatty acids. Those free fatty acids were then transported into the mitochondria and activated and converted in this fatty acyl-CoA molecule. And then that fatty acyl-CoA molecule is gonna undergo beta oxidation, where we're gonna produce this acetyl-CoA, as well as NADH and FADH2 molecules. And as you notice, we haven't actually produced any direct ATP within our beta oxidation. We've just produced this reducing equivalents NADH and FADH2, as well as acetyl-CoA. And these are going to be further metabolized in the pathways that we're going to cover for the rest of the lecture. And then you'll see how they actually produce ATP. So before we move on to the TCA cycle, we're going to briefly talk about rate-limiting enzymes and their importance within a metabolic pathway. So what a rate-limiting enzyme is an enzyme that catalyzes the slowest step within a metabolic reaction. So the speed of this reaction that is catalyzed by the rate-limiting enzyme is going to determine the speed of other reactions within our pathway. And so it's gonna affect the overall rate of the pathway. And again, we talk about some key enzymes in our metabolic pathways that we've covered. So our rate-limiting enzyme in glycolysis is phosphofructokinase. And again, this is because it's a slowest enzyme in that glycolytic pathway. So the activity of this enzyme, or the rate at which this enzyme catalyzes that reaction, is going to determine how quickly glycolysis can occur. And similarly, we talk about CPT1 being the rate-limiting enzyme in fatty acid metabolism. So the ability of CPT1 to get our free fatty acids into our mitochondria is going to determine the rate of how fast fatty acid metabolism is going to occur. And again, this slide is just showing you what we talk about in terms of beta oxidation, with CPT1 being the rate limiting enzyme and free fatty acid metabolism. So the ability of CPT1 to transport those free fatty acids into our mitochondria, it's going to determine how fast that free fatty acid is broken down. So again, the more free fatty acids we can get into our mitochondria through CPT1, the faster this beta oxidation process is going to be able to occur. So this is why CPT1 is considered the rate-limiting enzyme of fatty acid metabolism, because it dictates how much free fatty acids are transported into our mitochondria where they're metabolized. Similarly, in glycolysis, we have phosphofructokinase being the rate-limiting enzyme of this glycolytic pathway. So again, as we can see here, phosphofructokinase catalyzes that last step of the investment phase, or the conversion of fructose 6-phosphate into fructose 1,6-diphosphate. And so the rate at which this enzyme, phosphofructokinase, is going to be able to catalyze that conversion of fructose 6-phosphate into fructose 1,6-diphosphate, that's going to determine how fast this glycolytic pathway is going to occur. And again, this is because phosphofructokinase is the slowest enzyme within our glycolytic pathway. So this slide is just to basically help you further understand how a rate limiting enzyme works within a pathway. So here we have a given generic pathway as an example, and this pathway is composed of 3 enzymes. So here, as you can see, enzyme 1, 2, and 3, and underneath each enzyme, we're given their activities. So as you can see from the activities, is that enzyme 2 has the lowest activity in this metabolic pathway, meaning that the rate at which this enzyme is going to catalyze this conversion of these blue molecules into the green molecules is the slowest within this pathway. And so enzyme 2 is a rate limiting enzyme within this pathway, meaning that enzyme 2 is going to determine how fast this pathway occurs. And so even though if enzyme 1 is converting lots of these red molecules into these blue molecules, that doesn't really matter, because enzyme 2 is only able to convert a certain number of these blue molecules into these green molecules. And then similarly, even if enzyme 3 is able to convert lots of these green molecules in these black molecules, how many green molecules we have depends solely on the activity of this enzyme to you. And so basically, this example just shows you how a rate limiting enzyme controls a flux or flow through a metabolic pathway. So what we're going to look at for the remain part of this lecture is aerobic sources of ATP production. So aerobic, again, being the presence of oxygen. Specifically, we're going to look at the TCA or Krebs cycle, as well as the electron transport chain, as well as oxidative phosphorylation. And so basically, what we're going to look at is how these acetyl-CoA molecules as well as this NADH and FADH molecules that we've produced, that various steps of these metabolic pathways are going to be further broken down in order for us to produce lots of ATP. So what we're going to look at now is the tricarboxylic acid cycle, or the TCA cycle, that breaks down acetyl-CoA within the mitochondria. So again, this is an aerobic pathway, meaning that occurs in the presence of oxygen. And the TCA cycle is also known as the Krebs cycle, as well as the citric acid cycle. And this is again referring to the same thing. So as I mentioned, what the TCA cycle does is that it breaks down this acetyl-CoA that we've produced through beta oxidation, which we looked at earlier in this lecture, and also through the conversion of that pyruvate that was produced through glycolysis. So we have this, a bunch of acetyl-CoA molecules producing now, wanting to break them down further in order to produce ATP. In addition, the TCA cycle also produces carbon dioxide, a little bit of ATP, and again, more of these reducing equivalents. So NADH and FADH too. And again, as you're seeing, as we go through these metabolic pathways, we're always producing this NADH and FADH2 molecules. And you will see why they're so important later on when we cover the electron transport chain. So again, the TCA cycle is going to break down all the acetyl-CoA that we've produced from carbohydrate, as well as fat metabolism, as well as protein metabolism, which we'll look at later on this course. So here we have the TCA cycle. And essentially what it is, it's just a series of reactions that breaks down this acetyl-CoA molecule. So again, we have acetyl-CoA molecules that we've produced through beta oxidation, as well as that conversion of pyruvate into acetyl-CoA. And so once acetyl-CoA is gonna enter this TCA cycle, it's gonna be broken down through a series of steps. And we also produce a bunch of byproducts along the way. So you don't need to know every single step of the TCA cycle. What you do need to know is what's produced during each turn of the TCA cycle, as well as some key enzymes. So every time an acetyl-CoA molecule goes through the TCA cycle, we end up producing 3 NADH molecules shown here in the purple. We also produce 1 FADH2 molecule shown here in the light blue. And then we also produce some carbon dioxide. And then we also produce this molecule known as GTP. And GTP is essentially the equivalent of an ATP molecule. And then there's also 3 key enzymes that you need to know for the TCA cycle specifically. They're shown here in the bolded red font. So we have citrate synthase, which catalyzes the first step of the TCA cycle. So this conversion of oxaloacetate into citrate. Then we have isocitrate dehydrogenase, which catalyzes the conversion of isocitrate to alpha ketoglutarate. And this is the enzyme where we also produce this NADH and carbon dioxide. And then finally, the last key enzyme of acetyl-CoA is alpha ketoglutarate dehydrogenase. And then again, this converts that alpha ketoglutarate into succinyl-CoA. And then we also produce NADH and carbon dioxide at this reaction as well. And then SDH and MDH succinate dehydrogenase, as well as malate dehydrogenase. there are also additional enzymes that are involved in the TCA cycle. However, again, the key enzymes that you need to know are citrate synthase, isocitrate dehydrogenase, as well as alpha ketoglutarate